Hi everyone, welcome back to our tutorial series. In the last tutorial, we learned how to set up the Python virtual environment, install the Atlasapprox package, and run the first query. So today we will dive deeper and I am going to teach you how to explore gene expression data using the API. By the end of this session, you will be able to do this task on your own. Querying average gene expression data for single or multiple organs, identifying expression patterns of similar genes. Now let's get started. First, we need to ensure we have all the necessary packages installed for this tutorial. We will be using Atlasapprox, which is the main package for accessing gene expression data, and matplotlib to assist with visualizing the data later on. Now let's install them. If you are following along in a Jupyter notebook, you can run these commands directly in the cell. We use %pip install to ensure the packages are installed in the same environment that the notebook is running. After installing the required libraries, we need to import them into our Python script and then initialize the Atlasapprox API. Now that we have got everything set up, let's jump into the coding part. The Atlasapprox API covers data across 30 different species. Today, we will use humans as our example. The API has a handy method called average that lets us check the expression of specific genes across different cell types in an organ. For instance, if a researcher wants to know the average expression of some housekeeping genes they are interested in, say, in a human lung across different cell types, they can run the following code. This code returns a pandas data frame that shows the results. Let me break it down for you. Each row is a gene and each column is a cell type. The numbers in the table represent the average gene expression, measured in counts per 10,000 or CPTT for short. If you take a look at the data frame, you will notice that ACTB has much higher expression across all cell types compared to the other genes, especially in neutrophils. But looking at a big table of numbers can be tough to make sense of, so let's visualize the data to see the patterns more clearly. One way to visualize this data is through a heat map, which makes it clear and easier to see the differences in gene expression using colors. After running this, you'll get a heat map where the colors show the expression levels. Darker colors mean lower expression and brighter color mean higher expression. you can easy to see that ACTB stands out with high expression across all cell types, just like we noticed in the numbers. Now that we have seen how to check gene expression in a single organ, let's take it a step further and explore gene expression across multiple organs at the same time. We will use the same genes from our previous example and check their expression in 3 human organs: the blood, lung, and liver. Let's break this down step by step. First, we need to define a list of organs we want to analyze. Next, since the Atlasapprox API doesn't have a direct method to query multiple organs at once, we will use a for loop to go through each organ one by one. For each organ, we will call the average function just like we did before to get the average gene expression data, and then create a heat map to visualize the results. Now we have run the code. Let's take a look at the heat maps. The housekeeping gene ACTB is consistently highly expressed across all different cell types and organs, while GAPDH shows moderate but more variable expression. If you have other genes of all organs you want to explore, just follow the same pattern. Modify the features or organ list in the code above, and the API will return the data you need. Moving to the next part of this tutorial. Sometimes you might be interested in finding genes that behave similarly to a gene of interest. For example, to identify potential correlated genes or candidates for pathway analysis. The Atlasapprox API provides a convenient method called similar features. These can return the top genes with expression patterns most similar to a target gene in a given organ. Here's how to do it to find genes with similar expression to ACTB in a human lung. This returns a panda series, includes gene names, and the values show the similarity scores to the target gene. By default, similarity is measured using Pearson correlation, where a value closer to 1 indicates stronger similarity. To better understand how these similar genes behave across different cell types, we can visualize their average expression as a heat map. Here's how to do that. The heat map gives you a quick view of which cell types express these similar genes and how strong their expression is. We can clearly see the genes like ACTG1 and H3F3B show expression patterns very similar to the target gene ACTB. You can repeat this with any gene and organ, just change the feature and organ parameters in today's exemplar. Now you have learned how to query the average expression of genes across organs and cell types using the Atlasapprox API, and how to find genes with similar expression patterns to a gene of interest. That is for today. In the next tutorial, I will show you how to explore your cell type of interest in more detail and how to find marker genes that define specific cell populations. See you there!